Okay, Assalamualaikum and good evening everyone.
Hi.
So today we going to present about fish culture. Okay, next. Okay, this is our group member, Muhammad Syafa'i, Nur Sakinah, Amir, Faiz, Hisham and Najla. Okay, next. Okay, first of all, what is fish culture? Fish culture is where people own and look after a stock of fish. This might involve regular stocking, fertilization, and feeding, and also protection from predators and disease, and taking care of the environment. Okay, next. Okay, first, fish culture in natural waters is concerned with the replenishment and development of fish species in rivers, lakes, reservoirs, and seas. Fish culture is where you own the fish and care for them. Fish culture also becoming popular for food, employment, and for our income generation. Okay, next. Okay, this is an example of fish culture in China, which is they use the cage system. Next. Okay, the next example is fish culture in Kaptai Lake, Bangladesh. They also use cage system. Okay, that's all from me.
Okay, next I will present about broodstock. Okay, what is broodstock? Basically, broodstock is referred to a mature animal. In this field, fish used for breeding or fish multiplication process in aquaculture, which is fish farming. In aquaculture, we say a graphid female fish. Okay, for broodstock management, choosing species to use require consideration of the biology of the species. This include their size at maturity, method of reproduction, feeding behavior, and ability to tolerate adverse condition farm. It also consider whether they grow their own broodstock or obtain them from natural population. When natural population are excluded, the farm can be considered a self-sustaining unit independent for external genetic influence. Horn-reared broodstock are selected often as immature juvenile and grow out in suitable condition to sexual maturity. This animal requires stable water characteristic and a well-balanced, species-dependent, protein-rich diet. This enhances the germinal tissue for future seed stock as it forms in juvenile. The characteristic of the water in which the matured broodstock are held must be manipulated. The aquaculturist must consider the appropriate oxygen concentration, temperature, and pH of the water, all of which can be species specific. The feeding regime of broodstock in species specific and require consideration of timing and composition of the food. Protein, lipid and fatty acid composition is particularly important. The quantity of food intake can be altered into influence spawning and maturity. For example, low ration has been shown to reduce the number of fish reaching maturity while increase the fecundity of those which do. For those which do, when fry are desired, spawning can be induced in broodstock by manipulation of relevant environmental factor. In particular, the photoperiod can be altered into imply that is time to spawn a short photoperiod is known to advance spawning time, while the lengthened photoperiod can delay spawning. Artificial light can be used to change the apparent day length and indicate a different seasonal feature so as to delay spawning. Water temperature can be increased for the same purpose. Following spawning, the female brood fish are often stressed and they have lost weight. They require extra care and abundant feeding at this time to ensure survival to the next spawning season. So this is the brood stock selection, which is maturity of sexual organ, pairing or culture process, hereditary qualities. And next.
Okay, thank you Najla. So next I'm going to present about larvae. Alright, two pictures. These two pictures show the larvae of fish. The right one, there's a few types of larvae. Okay, next. Okay, what is larvae? Larvae is what we call we call each type for plankton. It is the newly hatched young of oviparous fish. Larvae also usually poorly formed but carry a large yolk sac, which is the yolk sac is for nourishment, and a very different in appearance from juvenile and adult specimen. Okay, for the larvae period in oviparous fish is relatively short, usually only several weeks, and larvae rapidly grow and change appearance and structure, which is a process term we call metamorphosis, like how before from worm become to butterfly, we call it as metamorphosis to become juveniles. Okay, during this transition, larvae must switch from their yolk sac to feeding on zooplankton prey, a process which depends on typically inadequate zooplankton density, starving many larvae. Okay, next. Okay, how larvae develop? Okay, the larvae develop from hatching until all fin rays are present and the growth of fish scale has started. We call it as squamation. Squamation is the state or condition of being scaly or scale. So, daripada larvae tu nak jadi ke ikan, dia punya sirip tu tumbuh daripada start pada ekor baru ke depan. Dia tak start daripada tumpuk-tumpuk ataupun daripada depan ke belakang. Okay, a key event is when the notochord associated with the tail fin on the ventral side of the spinal cord develop flexion, become flexible. Like I said before, dia start daripada fin yang kat belakang, baru dia tumbuh ke depan. Okay, next. Okay, larvae rearing. Okay, the larvae rearing is a typical intensive rearing technology ideally involving complete control over the environmental parameters and fish population. The first take place in a light environment using rotifers as first feeding and adding microalgae in the geared head Seabrim initial culture. The second protocol, frequently referred to as a French technique, which is applied for sea-based larvae rearing and is characterized by the dark environment during the first day after hatching and by the use of small newly hatched brine shrimp now play as first food. Okay, untuk kita nak buat larvae, nak ternak larvae ni dia ada dua cara selalunya guna. First cara adalah cara dalam cahaya yang banyak. Second cara adalah cara di mana tempat ternak tu dia gelap dulu untuk few days and then baru dia buka lampu after dia membesar sikit. Okay, next. Okay, ini adalah contoh untuk larvae rearing. Okay, the first picture untuk light rearing yang first punya cara. Second picture untuk yang second cara. Dia gelap dulu and then baru dibuka lampu. Yang ni dia dah buka lampu lah. Kalau tak buka lampu tak nampak lah gambar dia kan. Okay, yang gambar yang ketiga tu adalah cara macam mana dia ternak larva tu. First dia ambil daripada gamete and then baru dia jadi embryo and then baru jadi larvae. So lama-lama baru dia besar jadi ikan. Okay, next. Okay, for selection diet, apa yang larva akan makan sepanjang dia jadi larva? One should not only consider the impurities by alien particles but also the hygienic condition of the diet. Contamination of live food with bacteria is not necessarily hazardous but may have a tremendous impact on the microbial population in the associated culture medium and eventually in the fish or shrimps, flora, and consequently on the health status and the digestive capability of the larvae. A good diet should be readily available, cost-effective, simple as well as versatile in application. The persistent availability of sufficient quantities of food organism is of the utmost importance in continuously hatchery production. Untuk larvae ni dia memanglah makan benda-benda yang kecil, tapi kita kena pada masa yang sama sebagai peternak, kita kena jaga kebersihan pemakanan larvae ni lah, di samping kita kena cari yang murah. Okay, next. Okay, this is how we want, this is criteria how we want to choose for the food source of larvae. Okay, for the culturist, we have to find the availability, the cost effective, simplicity and versatility. For the predator, which is the larvae that kita tak ternak lah, yang larvae yang memang wujud sendiri tanpa peternakan. For physically, ada dua. First, physical and nutritional. For physical, they have to choose the purity, availability and acceptability. And for nutritional, they have to choose the food that have digestibility, energy requirements and nutrient requirements. Okay, next. Okay, next I will pass to my friend.
Okay, I continue with my presentation about grow out fish culture. There are type of grow out culture. Several type of to grow out culture system for freshwater have been developed in the southern. So there are three type of grow out culture. First part is concrete pond culture. Secondly, pen culture, or the other lakes. And third, cage culture. There is example for grow out. The location, there are pond, cage, and pen. Okay. Next slide. Okay, there are type of pond grow out management. There are nutrient renegation. Nutrient renegation is organic matter mineralized of conversion of nutrient. There are conversion nutrient by aerobic process. There is type organic matter mineralized of the conversion of some nutrient by aerobic is ester. Secondly is control of vegetation. Control of vegetation about weed that hamper fish culture. Fish culture that it can be operation. Dalam arti kata lain, dia membawa maksud control dia punya perjalanan punya kolam tersebut lah. Okay, after that disease control such a parasite. Kontrol daripada parasite ni membawa maksud mikroorganism yang boleh mematikan ikan dalam kolam tersebut. Okay, seterusnya pond maintenance. Pond maintenance is a part removal of excessive sediment in the pond bottom and facilitate. That mean pond maintenance mewakili pada dia punya pump dia sama ada boleh keluarkan oksigen atau tak, check dia punya kemampuan oksigen tersebut. Seterusnya fertilization. Exchange natural food productivity in the pond. Selalunya terjadi pada kolam tersebut dari segi filter tersebut. So filter, sama ada filter tu berfungsi atau tak. Dan fertilization ni adalah membantu pada proses pembesaran ikan tersebut lah. Seterusnya water quality management. Water quality management. Water quality, dissolve oxygen, pH. You grow out can check the pH at the pond and fish management. Dalam maksud lain, kualiti air ni dijaga dari segi oksigennya, pH-nya, pH-nya, pH air tersebut. Jika tidak diurus dengan baik akan membuatkan kematian pada ikan tersebut. Fish management pula dalam arti kata lain ialah proses menjaga stok ikan. Lagi satu dia punya berapa spesies yang ada dalam kolam tersebut untuk mengetahui dari segi pengeluaran ikan tersebut. After that, next slide. There is a pen grow out management in lakes. There are 4 parts. There is preparation prior to stocking, feeding, pen maintenance, harvest. It is same cage grow out of management in lake is same part pond grow out. They part maintenance of cage, harvest, pH, water level. They part same, but maintenance of cage is the different. Yang bawa maksud maintenance dalam sangkar ni, maksud maintenance dia berubah kerana dia dijaga oleh pengusaha ikan tersebut dari segi kualiti sangkar tersebut, sama ada rosak ke ataupun berlubang ke. So pengusaha tersebut perlu check maintenance daripada kes tersebut untuk mengelakkan ikan tersebut terkeluar ataupun sebagainya. Okay, next slide. Okay, I continue with my friend.
Okay, I will continue. For next slide is about breeding. Breeding is a sexual reproduction that produces offspring of larvae. There are 2 types of breeding in fish culture, which is natural spawning and induced spawning. Hello?
Yes.
Yes, we can hear you.
Next slide, Ella. Okay, for natural spawning. Natural spawning is males and females are placed together in breeding areas such as lakes or ponds so that they can spawn naturally. For natural spawning, the condition of water in lakes or pond also important to make sure the fish spawn naturally. The natural spawning also happens based on the type of fish, which is one is a seasonal, which the fish spawn at certain season such as rainy, flood, and spring, and non-seasonal, which is the type of speed. Okay, next. Okay, one of example natural spawning is which is tilapia, Oreochromis species. The eggs of tilapia fertilized by a male. The female we collect and incubate the eggs in the mouth. And the egg we hatch about 5 to 7 days. Okay, next. Next adalah hello. Okay. Okay. Next. Before this. Belum tu.
Ke belakang tu, ke belakang lagi. Line page number. Yang ni. Depan. After example. Dah saya.
Ah. Aduh.
Depan satu lagi.
Ah.
Induce.
Induce spawning. Aku slow.
Ni.
Yang ni.
Clear tak aku cakap ni?
Okay.
Clear.
Laju sikit.
Okay. Next is induce spawning. The induce spawning is one of the common methods that to stimulate the ovulation of the fish in the hatchery, commonly for species that not reproduce in captivity, which means for the species that cannot in natural spawning. So we make the induced spawning. The main things to make induced spawning is to supply fish outside the normal spawning season, and it's very important for intensive culture. It also develop superior strains through selective breeding and hybridization method. So hypophysation, which is the process of inducing fish to spawn through the use of hormone. Okay, next. Okay, induced spawning. The important aspect for induced spawning is one is environmental, which is the temperature, water volume, pH, and salinity. This aspect must be suitable for fish to breeding. Second is hormone for induction, which is human chorionic gonadotropin, luteinizing hormone releasing hormone, gonadotropin releasing hormone, follicle stimulating hormone, and ovaprene, ovaplan, and ovatide. The most commonly used is ovaprene and ovatide. The criteria of male and female broodstock, which is must large, productive, healthy, and free from disease. Okay, next. Okay, this is example step for African catfish breeding. First is we must identify and separate the sexes, which is a female have a swollen genital open, while the male is genital papilla. You can see at the picture. Next is we can select and check the female fish with ripe eggs and weight the female. Third one is prepare. You can see we inject at the atas belakang ikan tu. Inject ovaprene ataupun ovatide. Okay, next. Okay, step keempat adalah we inject the fish and wait for 10 to 12 hours. After that, we press the egg out of the female fish. Kita tekan, kalau tengok kat gambar tu, dia picit je kat perut ikan tu, keluar telur banyak. Okay, step keenam adalah we bring the male out and kill it. After that, we remove the nutsack. Okay, dan dalam gambar, nampak kita gunting dia punya apa ni? Kita cut the testic male into the beak to release the sperm. Okay, after that We pour the sperm and female egg in bowl slowly and spread the egg on spawning sponge inside incubator. Number 9, and lastly, we will wait until the egg hatch and come. It takes commonly 20 to 36 hours. Okay, next.
Okay. In conclusion, we know fish culture is very important to our nation because it can give more income and save the world from any disease. For example, is because it is very important each other to save our environment and to the income. Like I say, for every people in the behind the lake or fish culture. Okay, lastly, fish culture is very important to secure the livestock and always supply of food for our nation and all human in this world. Okay, thank you for all about this present. Thank you.
Alright, thank you.